Test de son, vous m'entendez?
Oui.
Et vous voyez l'écran complet?
Oui.
Parfait. So hello everyone for this webinar. We're just gonna wait another 2 minutes for everybody to join the webinar, and we'll be back soon. Thank you. Thank you all for joining this webinar. We're going to wait another minute before we start it. Welcome everyone to this webinar organized by WSP and FREDSense. The title of this webinar is On-Site PFAS Detection in Hours, Real-World Lessons from WSP and FREDSense. Just some housekeeping instruction. The webinar will last 30 minutes with a good 15 minutes period for question and answer. Please mute yourself during the presentation and send your question in the chat box during the presentation so we can answer them at the end of the presentation. So there's a Q&A box, so this is the one to use, not the chat box just beside. Also, for the questions that were sent prior to the webinar, they will be answered as well at the end of the presentation. So today, you have me as your moderator and the two presenters, Hamza Alouane and Julia Guéguena. Myself, I'm from WSP. I'm a chemical engineer with 17 years experience in wastewater and drinking water treatment. I develop and design systems for community, ships, military base, school, mines, food industry, rendering plants, basically most of the industries. I'm a subject matter expert in electrochemical processes and led the development of the P-phaser system that we will briefly discuss today. I author 4 patents, 1 patent pending, and more than 20 peer-reviewed papers, conference, and proceedings. So I'll be your moderator today. As presenter, we have Hamza Alouane, which is an environmental engineer with 8 years experience in water-related projects. He specializes in the design, operation, and maintenance of water treatment units. Mr. Alouane also has a major experience in project managing. He graduated from ETS in Montreal in 2016 and joined WSP Canada in the Industrial Wastewater Treatment team in November 2023. So welcome, Hamza. And Julia Guéguena from the company FREDSense. Julia is an experienced biochemist, microbiologist specializing in RNA research and sensor development. Currently, leading the FRED-PFAS product initiative at FREDsense Technologies, she brings over a decade of academic and industry expertise in environmental diagnostics and synthetic biology. Welcome today, our presenters.
Valérie, I'm just going to step in just a second because you're showing like your full screen. So on voit aussi comme la diapositive suivante puis les notes. So perhaps
like this is better?
Yeah. Oh,
Yes.
absolutely. Thank you.
Perfect. Yeah, I wanted to know if it was the good switch. Good. So we're recapping. So today, the presentation is all about a collaborative project. It started in March 2024 when WSP and FREDSense started a project to test the FREDSense kit to measure total PFAS in groundwater and treated groundwater. It's a 4-phase project. We started with lab test, then pilot test, then field kit optimization and deployment. The objective was to test the FREDsense, the FRED-PFAS accuracy, uncertainty, matrix interferences, turnaround time without hours, and especially the usability. Tests were done on groundwater, electro-oxidation, and granular activated carbon treated water from an airport and an oil chemical plant. This webinar presents the result of this collaboration. The agenda will be first to describe a bit the PFAS analytical toolbox, the problem of non-dynamic PFAS remediation, solution, which is a real-time PFAS analysis. Then FREDsense will present the FRED-PFAS technology. WSP will present lab-scale use of the FRED-PFAS technology, and then the use at the pilot scale of this technology. And finally, we will tell you what are the benefits we see in this technology and obviously some lesson learned during the use of this technology. So without further ado, I will introduce you to Julia, who will start the presentation.
Thank you, Valerie. So let's begin with the PFAS Analytics Toolbox. And this iceberg graphic says it all. PFAS represent a vast and chemically diverse family. We're talking about 10,000 known compounds, but our ability to measure them is still catching up. Most of the time, we rely on targeted methods that focus on a very small slice, around 30 to 40 PFAS. These are the compounds we can accurately detect using validated lab technique around LCMS like EPA 1633 or EPA 537. Beyond that, we have non-targeted approach, which aim to capture a broader picture, like the total fluorine content with TOF or the precursors with TOP. But these methods are still developing, more difficult to interpret, and not yet widely adopted. So when we report PFAS levels, we are looking at just the visible tip of a much larger iceberg. The vast majority remain unseen, unmeasured, yet potentially just as relevant. And since all of this work happens in specialized lab, it's resource intensive and slow, representing a major constraint for timely decision-making in public health, environmental monitoring, and policy. The EPA introduced this triad approach to improve site remediation. It's built on 3 pillars: a systematic planning, a dynamic work strategy, and real-time measurement. In principle, it's designed to make cleanup faster, smarter, and more cost-effective. But when it comes to PFAS, we're missing a crucial leg of this triangle: real-time measurement. Without the real-time data, we're stuck in a non-dynamic approach. We collect the sample, send them to the lab, and then wait, sometimes weeks, before we can decide what to do next. This delay breaks the triad. It forces us into a stop-start cycle, causes inefficiency, and drive up cost and timeline. Hamza will be able to provide you an example in a couple of slides there. While the triad is a great model, in practice, it's currently not fully achievable for PFAS, simply because real-time monitoring tools aren't available yet. Now, imagine if we could screen for PFAS on-site and get results the same day. That's exactly what a tool like FRED-PFAS enable. And just to be clear, this doesn't replace lab testing. Labs are still essential for confirmation, compliance, and detailed analysis. What FRED-PFAS offers is a complementary frontline tool that brings speed and agility to the field. Instead of waiting weeks or months for lab results, team can start making real-time decisions on-site the same day. This creates a truly dynamic workflow, just like the EPA triad approach envisioned, and it breaks the stop-start cycle that slowed down PFAS projects. The results: faster timeline, smarter decision in the moment, reduced project cost, and more efficient use of lab resources by only sending in the most critical samples. In short, FRED-PFAS could be the missing leg of a stool, the tool that can finally bring the triad to life for PFAS remediation. So what exactly is FRED-PFAS? FRED-PFAS is a portable screen tool designed to bring PFAS detection directly to the field. It's fast, simple, and rugged. The system is made up of 2 core components, as you can see on the left part of your screen. You have a FRED capture unit, that house the entire system where PFAS are extracted from the sample, and a detector, also called FRED-Fluor, that allows you to measure a proprietary biosensor technology. To operate it, all you need is a reagent pack, which include the required plastic labware and our FREDSense proprietary blend, everything being pre-packed and ready for field use. The whole system is housed in an impact-resistant, waterproof case and weight of just over 5 kilograms, which is about 12 pounds, which make it truly field-capable and briefcase-sized. We designed it with simplicity in mind. It's a 5-step process, color-coded and numbered, and most importantly, it's designed specifically for consultants and operators. You do not need a specialized lab training to operate it. You can run 2 samples at the same time, and it delivers results in under 4 hours instead of weeks when sending it to the lab. And because sometimes you need to run more than 2 samples as fast as possible, we can stagger the sample up to 16 in a day and measure them all at once. Most importantly, and the most crucial question that everyone is wondering is, what does FRED-PFAS measures? It produced a trend that aligned with our third-party data, and to answer this question properly, I would like to bring back the iceberg diagram. So let's revisit the PFAS iceberg that we saw earlier, where most of what's out there remain hidden below the surface. FRED-PFAS help us to see more of this hidden mass. While it's not a targeted method that identifies specific compounds like EPA 1633 method would do, it functions more like a total PFAS screening tool. It captures a broader signal of PFAS presence in a sample. So it's a non-targeted, but also highly selective. It's tuned specifically to detect the fluorocarbon backbone that is characteristic of PFAS molecule. It's able to detect C5 plus PFAS, including many precursors, which are often missed with targeted methods. In terms of performance, FRED-PFAS can reliably detect down to 1,000 nanogram per liter, or 1 part per billion, in real-world sample, not only in clean lab water. It works well across a variety of matrices, from AFFF contaminated site to industrial wastewaters. So while we are not seeing the full 10,000 compound universe, FRED-PFAS gives us a broader, faster, and more field-ready view, helping us to close the visibility gap. We've done a lot of work to really understand and characterize what is the FRED-PFAS sensor detecting, but most importantly, what is not detecting. So the graphs here shows you the thorough testing across a wide range of individual PFAS analyte, as well as AFFF mixture. You can see that we looked at a C8, so PFOA, PFOS, C6, PFHXA and PFHXS, or C9 with PFNA, and shorter chain compounds like PFBS and PFBA, or less known or less worrying about 6,2-FTS and PFTDEA. These results reflect the sensor performance specifically and not necessarily the full system output, as the overall detection depends on a capture step, which can be customized based on project requirement. We've also screened our FRED-PFAS sensor for potential interferences or cross-reactivity using compounds commonly found in various type water. The goal is to make sure that the signal we are seeing is truly PFAS related. We've tested common substances found in groundwater, like humic acid, total hardness, or salinity, but also some less typical ones like benzene and octane from hydrocarbon and petroleum site, or SDS and octanoic acid. And you can see that compared to our PFOA, our sensor is not picking up any of these components. And this is not a one and done process. Our team is continuously expanding this data set. We actively working to test new compounds, real-world sample, and interference scenario to further refine the sensor profile. It's really an ongoing project with a clear goal to provide the user with reliable, field-ready insight that they can trust, backed with real data and transparent testing. So here's a quick look of the FRED-PFAS development roadmap, showing where we are today and where we are headed. Right now, we are detecting at a ppb level with proven performance in AFFF impacted sample, groundwater, and industrial matrices. Over the coming year, we are pushing towards a lower detection limit and then provide a broader matrix type like soil and biosolid in 2027. In between this big milestone, we will also be improving the user experience of the product, the hands-on time, and the overall product. So that's an open call there. If you're interested in participating in any pilot project and or supporting any of the development, we would love to connect and have your input on that. So as Valerie mentioned earlier, we want not only to show you what a FRED-PFAS is, but also how we had the opportunity to put it to the test in the real world. And we've been incredibly fortunate to do that in collaboration with WSP, a recognized leader in PFAS science and remediation. The team brought a deep technical expertise and forward-thinking mindset, which made them the perfect partner to help validate this technology. So the process started with a proof of concept phase to see how FRED-PFAS would perform in WSP sample type. In phase 1, we have used a high concentrated sample from groundwater site. From there, we moved on into confirming the limit of detection by using a mix of single-digit ppb and ppt sample. And the next phase is, in my opinion, the most exciting one is the testing FREDsense performance in the end of WSP own staff with our team providing support needed during deployment. Now, let's take a look at the first results from a collaboration with WSP. And Valerie, if you can go on the next slide. So this first round of testing was split into 2 phases. The phase 1A focused on a sample with higher PFAS concentration. The graph on the right is showing you the results obtained for this one sample using 2 targeted methods, EPA 537 and EPA 1633, and 2 non-targeted methods, FRED-PFAS and TOF by CIC. What we saw and is presented here was really promising, with FRED-PFAS consistently detected PFAS in line with both a TOF assay and an EPA 1633. We were also able to streamline the workflow by using reduced sample volume, which means less processing time and a faster turnaround overall, which is a big win for field efficiency. In phase 1B, we looked at lower concentration sample, closer to the detection limit of the FRED-PFAS system. Here, we validated our 1 part per billion detection limit using a 1.3 ppb sample, also showing an especially stronger analytical confidence when sample volumes were increased from 500 mL to 1 liter. That said, sample with 730 nanogram per liter and 15 nanogram per liter based on EPA 1633 were far below our detection threshold and were shown as non-detect. This really helped us to understand both where the FRED-PFAS performed well and where we needed to refine further to improve the reproducibility. And now I'll pass it to Hamza to show you the results for phase 2.
Thank you, Julia.
Mm-hmm.
So if we can go to the next slide, Valerie, please. So WSP developed 2 years ago the P-Phaser, which is a system using electrooxidation for PFAS destruction, GEC for trace PFAS removal, and also equipped with an integrated perchlorate removal process. For the BDD, it's a durable boron doped diamond electrodes, also that we use in a full-scale designed water treatment unit that we incorporated, for example, like 6 years ago in one of our projects, and is still running and no changes are required for those electrodes. The equipment of the P-Phaser are built in a marine container that can be shipped to any site. And during the pilot study, some of the variables needs to be fixed, such as EU treatment time that can go from 15 minutes, for example, up to an hour or sometimes more than that, and also the chemical dosing to get the optimal PFAS removal rates. And those variables usually depends on the water matrices. For example, we had a pilot study last year in a chemical plant, and we stayed for 2 months on a site. But 80% of those 2 months were only waiting for the PFAS results to be received from the labs. For example, it takes up to 10 days to get the results from the lab and also for a high cost. This led to a staff demobilization each time and also being in a standby situation until receiving the results. Having the FRED-PFAS at that time could have reduced the cost and also made the pilot study duration only 1 week. Some of the challenges that we face with the lab analysis is that the lab control sample recovery is either above or below the data quality objectives, which means that the PFAS concentrations are either overestimated or underestimated. And this was the case for 22 analyses out of 22 that we did during the pilot study. So the current analytical technologies do not allow such perfect accuracy as for pre-established parameters that we use usually in our industry. So last year, the FREDsense crew came to Montreal in order to test the FRED-PFAS in our facilities using the water that we collected from previous projects of WSP. For example, the first sample that we analyzed was a high PFAS concentration water coming from an airport groundwater. So the FRED-PFAS correlated very closely with both EPA 1633 and TOF for real-world groundwater ppm level samples. As we can see in the graph at the left, we tested the water with the FRED-PFAS and also we duplicate this with the EPA 1633 and the TOF, and we can see that the three results are almost around the 7 ppm. And also we recorded a high repeatability for the FRED-PFAS analysis. For the second sample that we analyzed was the low PFAS concentrations coming for groundwater coming from a chemical plant. The sample were at concentration near the both methods detection limits, 2 ppb for the TOF and 1 ppb for the FRED-PFAS, which results in a higher potential error. We can see on the graph that the TOF on the right one, that the TOF error is further pronounced when comparing the raw water with the EU treated water, and the result should have been the same. So finally, the FRED-PFAS trended with both EPA 1633 and the TOF as expected, despite the variability between the measurements. And also, finally, the EU treated water plus GEC results were non-detect since the water was already treated by the P-phaser with the GEC and the EU. So for faster PFAS remediation, now we have two FRED-PFAS devices in Montreal WSP Treatability Center, and also we're able to do lab scale electrooxidation study in weeks using the FRED-PFAS compared to months without the FRED-PFAS. The lab scale electrooxidation is a similar process to the P-phaser, but for small volumes of water. We can treat up to 700 mLs once we receive the water samples from our clients. And also with the P-phaser, we can treat up to 9.6 cubic meter per day, combined with the FRED-PFAS for few weeks pilot projects at a reasonable costs. And finally, the FRED-PFAS alone can be used on site for same-day results, which gives us the ability to react quickly in an emergency. For example, a client had a spill a few weeks ago in the pipes leading to its treatment plants, and it was crucial for us to determine quickly the PFAS concentration and its evolution in the system at different points, which was possible using the FRED-PFAS. So some of the lessons learned today are the FRED-PFAS is easy to use and can provide fast results within 2 to 4 hours. The FRED-PFAS can detect total PFAS at 1 ppb limit of detection for now. 2 duplicates are usually recommended with the FRED-PFAS in order to confirm the measurement uncertainty, and both of them can be completed in 2 to 4 hours since the machine can take 2 samples at a time. And finally, the FRED-PFAS did not suffer any water matrix interferences in tested groundwater, EU water or GEC treated waters. Thank you for your time. And now we're open for questions.
So thank you so much, Hamza and Julia. It was very informative. I hope that our audience appreciated the detail of the experiments and the limit of the technology and its benefit. So now we're going to open the ground for the questions. And to hit the ground, there were a few questions that were asked by the audience before the webinar. So I will ask them, and our presenter will be able to answer them. So let's start with the first question that was asked. How many test samples were used to build and validate the FREDsense PFAS detection system using the standard lab methods? So, Julia, the question is for you.
Yes, absolutely. So the FREDsense PFAS detection system underwent quite a bit of development and validation. Using a wide array of samples, we have both used synthetic standards and actual environmental sample, some like groundwater, AFFF. The overall validation process involved well over 100 samples and comparing the results with EPA 1633 and TOF each time to guarantee the accuracy, reliability, and consistency across a diverse sample type. Additionally, we have in-house and LCMS capabilities, so it's enabled us to conduct daily comparative tests using both 1633 and TOP methodologies.
Thank you. So now the second question, what is the detection limit of the FRED-PFAS system? Can it also detect other compounds and physicochemical experiments that may be present in the sample? Julia?
So the FRED-PFAS system generally achieve detection limit in the low ppb range. We anticipate launching a product with PPT detection capabilities in early 2026 now. Specifically within the first quarter of the year. That being said, the precise detection limit can fluctuate based on matrix and specific PFAS compounds. So for more detailed discussion on like the timeline and the exact specification relevant to each application, it will be easier to follow up by email.
Very good. Question number 3, what is the calibration process of the FRED-PFAS system?
So the calibration process is fairly simple. We are providing all the calibration solution that you would need, and it's provided in the user manual, and it takes about 10 minutes. We recommend to do it currently every week, and we are improving this system to get to over a month.
Thank you. Number 4, is there a method available for analyzing PFAS contamination in soil samples? How does FREDsense ensure rapid and efficient testing given the challenges of sample preparation and extraction in these matrices?
Currently, the FRED-PFAS is only configured for testing on water samples. Our R&D team is investigating soil-based PFAS detection with potential application target in 2027. So I don't have more information on that part. We will 100% do all the tests and can provide all the information to ensure the efficiency of the testing.
Very good. How robust is the technology?
It's pretty robust. The FRED-PFAS capture and detector are housed in the rugged nano cases, so the operation only requires stable flat surface, avoid direct sunlight to prevent overheating and potential results interferences. But that's pretty much all you need.
Okay, and we did talk about the metric interferences, so we're not going to ask another answer for that. And the last question, is there a field screening option for PFAS that offer faster results? How do the results compare to the lab analysis at the 1633? And can the system detect PFAS at concentration below the Health Canada drinking water limits? What is the availability and cost of the field screening system? So all yours, Julia.
Lots of questions. The FRED-PFAS provides right now results in 4 hours, potentially closer to 3 hours when the fewer volume of sample, if the concentration are high enough. I think it's an open question there is how fast do they need to be, because we can absolutely discuss that. Unlike targeted method like EPA 1633, for the next question, FRED-PFAS detect a broader range of PFAS and offers more of a total PFAS concentration value rather than individual species. So a FRED-PFAS typically trend higher than EPA 1633 and closer to a TOP or TOF analysis. For the Canada system detect PFAS concentration at Health Canada drinking water limit. So currently, if I'm not mistaken, the Health Canada drinking water limit is suggesting a total PFAS concentration at 30 ppt. With a 1 ppb detection limit, FRED-PFAS does not meet yet this standard. However, all of our efforts are underway to lower this detection limit for early 2026. And finally, for availability-wise, we're currently selling into September 2025. And for the pricing, it would depend on the order size and all of that. So we'll be more than happy to discuss in the oncoming project by email or let's set up a chat.
Thank you, Julia, very much. So now I'm going to switch to Q&A questions. So from the chat, what is the level of efforts for the operator? So that's going to be for Julia. During a 2 to 4 hours turnaround time, is the unit operator actively working on using the unit or just, just a minute, I'm going to post away this, or just waiting for results and able to work on the task? Actually, I'm going to target the question to Hamza, which was the operator in this project. So what do you do during the 2 to 4 hours of turnaround time with the tool, you know?
So what is the question?
So for the operator, it takes 2 to 4 hours. Are you just waiting, looking at it all the time, or do you do something else? So they want to know this.
You can do other stuff during the process, especially during when we're heating the sample. But usually during the first hour, you're actively working on the machine. But you can do some other stuff at the last periods of the testing.
Perfect. What are the consumables per sample cost for the field application, Julia?
Sorry, can you repeat the question?
What are the consumables per sample cost for field application?
Currently, it's about 100 USD per test kit, so reagent pack. And yeah, after that, it also will depend on the amount ordered. And for all the different kits, we usually provide, everything is packed in for 2 tests. So usually, because we usually suggest to do 2 tests at the same time. So
it's about $200 to get the results within 4 hours, roughly?
Yes.
Matt, Rick, so please provide resource with whom I may discuss PFASer. I have a site requiring design of a water treatment system. So Rick, at the end of the webinar, we're going to take the name and email of everybody, and we'll take the question and we'll be able to send it to you, but it's going to be myself or Hamza that's going to answer your question. Karina Chan, thank you for the presentation. How does a FRED-PFAS detect PFAS? Aha, on the science, Julia. How does it detect?
It is, I cannot really tell you everything that is in there. It is quite proprietary, and I cannot give you all the secrets right away. The technology is you have a polymer that will bind some of the PFAS, and there is also a fluorescent molecule involved in this whole process.
Okay. Russell Garrett, what are typical equipment costs? Are spec sheets available? So this, I'm assuming, is on the FRED-PFAS product, Mr. Garrett, I'm assuming. If not, just say no when we switch to the PFASer, but you can answer Julia about your cost.
I think both of them we can have the answer for both of them. The FRED-PFAS cost is going to depend on the application. We have product spec sheet that we can send as follow-up email for that part.
Perfect. So we can reach out to Julia or FREDsense directly for that question. Olivier Hamon, what is the volume required per sample for the FRED-PFAS system?
Currently, we're suggesting 500 mL for samples. If you're getting closer to the limit of detection of the current one, so 1 ppb, we're suggesting to use 1 liter to really step away from this uncertainty zone and get more accurate results. And it can also be up to like 100 ml if your sample is quite concentrated.
A question from Mr. Chavan. Can the system be used by non-lab personnel with limited technical training? So that would be my first question. Yeah.
Yes, it's the goal is to really have everyone being able to do a PFAS analysis on site and really know.
So me, I'm a project director. I did the test. I use it. AMSA use it, and then we had a technician and a junior engineer. So we wanted to see if the test, the way we use it, would be different, and everybody followed the 5 steps, and we got to the same results. So I'm happy to say that at different level in a consultant company, you can do that. How does a FRED-PFAS system differentiate between total PFAS and PFAS precursor in complex matrices like landfill leachate or industrial effluent? So that's the question here.
So currently our system is not designed for landfill leachate or more complex samples. We are actively working on getting this very complex matrix in-house. So if whoever asked a question is interested in helping us figure that one out, it's quite complex because this type of matrix are changing depending on the site, depending on the day. So it's really complex. So I don't have an answer for you just yet. Next webinar.
It's like a turbidity meter. Turbidity meter is as good as the matrix of the water you're measuring. The turbidity is affected by so many things, so it's really about learning the tool. What is the approximate cost per test, and how does it compare to lab-based testing, including TUF?
That would be more a question for our BD team. I don't have the exact price analysis there. We have a tool that we can provide to really break down all the different analysis and what is the advantage of using a FRED-PFAS versus another method. The kit itself, like the reagent that needed is 100 USD. So to do duplicates, you need 200 USD, and a TOF analysis, depending on which site you're using and which company, is about the same price, but you would have to also wait and send the sample away.
Yeah, that's the main advantage. And also the detection limit is 1 ppb for you, while the TOF, if I'm not mistaken, is still 2 ppb. So that's an advantage. And the fact that you get the result in 4 hours instead of 3 days, even though you accelerated more for the TOF. So that are the advantage, I would say, for the FREDsense compared to the TOF. What are the biggest current limitation or area under development for the FREDsense?
Can you repeat the question?
What are the biggest current limitation or areas under development for FRED-PFAS?
The biggest limitation right now that we are seeing is really like the detection limit that we are actively working on. So we are aware that 1 ppb is not enough for a lot of application. We also working on really characterizing what is the sensor seeing. So it's we're not claiming to be a total PFAS. We know there is some species that are missed, like the ultra short chain. So we really working on getting this part done. So that's one of the limitations. We're really trying to understand what is exactly detecting and the limit of it.
Okay. A question from Nathan Angelin. I'm sure you will cover these topics. I have to drop. Does WSP have a preferred arrangement with FREDsense as a result of this collaboration? Have you set a price for the FREDsense unit? Do you rent the unit? Last question. FREDsense received regulatory approval as an acceptable alternative method. So for the first part, it's a collaborative agreement, and we got 2 units out of this at a certain price. So we're very happy because of the time WSP give. For the other one, the price of the FREDsense unit, that's going to be something that Julia can discuss offline. But I guess this one is technical. As FREDsense received regulatory approval as an acceptable alternative method?
This I'm not sure if it's comparing with different EPA 1633 or other methods. We do compare all of the results as much as possible with EPA 1633 and top. We have capabilities in-house to do it, and we also send some sample for third-party analysis to really confirm the concentration and all the different measurements that we are seeing.
Us as a user, WSP, we will not claim a certain treatment performance only on FREDsense. So what we're going to do is use a FREDsense kit during a trial to fine-tune the system, and then at the end of the trial, you send the lab test because they're official, recommended by the provinces or the federal, and then we finalize a report with the accredited lab certificate. But in the middle, we can use FREDsense because we don't need to have always lab that are accredited. So that's how you can reuse the FREDsense. And the last question here from Samir Nevi. Has biochar been used for PFAS adsorption? There are studies that show that it works.
I'll take that one. So for the biochar is an old appellation of GAC. It's already working, but for the P-phaser, for example, we're using the EU destruction. For the biochar or GEC, we're just moving the PFAS from the water to the carbon that needs to be disposed after. But in our case, for the P-phaser, we're using the destruction, using the electro-oxidation, and just for a final tuning of the treatment with the carbon.
Very good. So, okay, so now,
No,
Vincent, yeah?
Valérie, yeah, there's an extra question that has been asked on the Q&A. So if you just scroll up, there's going to be
Scroll up. Okay, they're
4 or
by
5
order
questions.
of,
Yeah.
okay, so 11 minutes ago, they are there. Okay, so let's move on with that. What is it? No, this we've done already. So this one here, it's
Just go up, and then go down.
okay.
I
Up,
can
up.
I can ask it.
Yep.
Yeah, go for it.
So how does the calibration look like? Can it be done by non-technician?
Well, I can answer the question. I can also have Amza doing it because I believe he done it not that long ago. It's Amza. Is it really complicated?
Yeah,
Let's go.
yeah, it's pretty straight ahead. You receive solutions that you put in a machine, and you just follow the steps for the solution. I think it's from solution 8 to 1, one after the other one, and then you just get the calibration done. I think it takes up to 10 minutes to get all the solutions in the machine. So it's pretty easy.
Okay.
That's all.
Other question from Mike Eversman. How do FREDsense results compare to top assay results?
Great question. We are comparing actively right now with our own top. So we're testing exactly what we're measuring. It looks like we are picking some of the precursors, not all of them, because that would be too perfect. So it's fairly similar to a top. It does detect some compounds that not necessarily detect with top, or that the other way around. So it's fairly comparable. We can potentially have some information to show you by email to follow up and maybe show you more of the really detailed results of that.
Thank you. I think we answered the other one, but there's another one from Dan. Is it recommended to run duplicates of each sample?
It depends on the application. Honestly, if some of the samples are really high concentration, if you need to know how high it is and really precisely, yes, we recommend it to do it. If it's just a washing, for example, from AFFF rinsate, if you just want to start washing and you know it's really high, we don't really recommend to do all the duplicates. We suggest to do more of the final sample, to do more duplicate to confirm all the cleaning process is done. So it depends on the application. We can provide guidelines depending on what you've been testing.
Thank you. One from Tom. The comparative results illustrated during the webinar all had 1,633 results that were the same order of magnitude as TOF results. In practice, we often see 1,633 less than 1% of TOF in our AFFF applications. How does FRED-PFAS perform under these conditions?
That's a great question. I think I would like to take a bit of time to answer this question, so maybe we can follow up by email on that part, so I really get a thorough answer on that part, if that works for you, Tom.
I have a few questions here that were on the chat, Vincent. A very long one from Russ, Jan Russ. So
go ahead.
Okay, good. So basically, is very knowledgeable. He said that the FREDsense kit cannot detect. Do you guys see the chat, or I'm going to read slowly? The FRED-PFAS kit cannot detect most short chain and all ultra short chain PFAS, which are transformation product of the electrochemical oxidation. So looks like detectable PFAS could have been converted into other short chain PFAS by the ECHO. I'm assuming he's talking about electrooxidation, which the FRED-PFAS cannot detect, and so they are discharged. So I'm just going to stop here. So for sure, we need to comply with regulation, the applicable regulation at every site. So if we use FRED-sense for the electrooxidation system, it's really to optimize it. So often the electrooxidation system, the PFASer, we bring PFAS from ppm to ppb, or ppb to ppt, or ppm to ppt into the cost-effective way. We use a FRED-sense to see the curve of reduction of the EO. And we always send the raw water sample to an external lab, as well as the final treated water to an external lab to compare how it behaves with the FRED-sense. And only when we like that there is an equivalence, we continue using FRED-sense without the analytical lab. We will never rely on the PFAS alone for the PFAS kit, the FRED-PFAS kit alone at the moment. We need to gain confidence, but it helps us tune the system very quickly in the middle of the development of the system while you don't need to wait for the lab analysis. So this is really the value of the system, a bit like the ACH, the ACH spectrophotometer. We use it for COD within 2 hours. We use it for TOC in 2 hours. So it's a very rapid way of tuning a system. And then you certify it with analytical lab results. So that's a bit for this first part of this question. And then the second part is no attempt to test the FRED-PFAS kit against target ultra shorts. So that's a question in itself, Julia.
I believe Melanie answered some of the question there. So we're testing all the available standard, and many standards are not necessarily available for certain PFAS compounds. So the work is ongoing for a lot of this ultra short chain that are often lost, and a lot of them are also being lost in the SPE process. So the first step of the capture. So yeah, we can further optimize it. So I think
so the person is saying that you cannot use it for how can anyone rely on the data that detect the PFAS when comprehensive assessment of the PFAS is not possible? This is what you're answering. You're working on developing it, and right now is a screening tools in the PPB detection limit, and you always compare against a lab. No comparison of FRED-PFAS against multiple comprehensive NT approaches. I've published on detection of 1,000. So it's a long text, but Julia, you can read it here. But it's come to the same thing. You're doing this work, and now is available for certain application, not all, obviously, and not to claim compliance with regulation at the moment.
Yeah, and absolutely, we're not claiming to be a total PFAS sensor like a TOF would do. It's just a tool to do in-between analysis there and like we said, there is more than 8 million PFAS species. So we are slowly catching up on this 6 million and trying to get all of the different compounds detected. So it's really a work in progress.
And same thing as the analytical lab method. They don't detect them all, right? So it's progressing, I guess, the technology. Okay. How does FRED-sense compare to top assay analytical results? Oh, the top this time,
Valérie,
TOP. Yep.
No, all those questions were answered. So everything that was in the chat was in the Q&A.
This one was not answered. It's this one I didn't ask it. The TOP.
I did ask it to how does FRED-sense compare to top assay analytical results?
Okay, sorry about that.
Yeah.
Okay,
no
so it's all done now?
Yeah.
All those are good. Okay. And no other one in the Q&A on the other side?
Not yet.
Okay, so we're going to wait 2 more minutes. It's 12:48. And after that, if there's no more questions, we will close the session so that everybody has time to have a lunch. Okay. So I guess
I believe we'll be sending the presentation.
Oh, there's a person here that raised her hand. So Vincent, can we take?
So there's another question in the Q&A. So what are the potential analytical inhibitors during analysis? That is compounds.
So I think that would refer to the interferences that we would be testing. Currently, we have found that a high level of some of the solvent and some detergent will impact the detection. We also been finding that some, I believe it's iron, that is also could be an issue with a detector. So we're still processing all of that. We're finding all of them. And once we find it, it's a whole different process, because once we found that there is an interferences, we have to look for method of taking care of these interferences and removing them. So it's a lot of pre-treatment happening probably.
But there's one last question from Bailey. Is there a list of the PFAS the kit can detect? In comparison, 1633 can screen for 40 kinds of PFAS.
Yes, we do have a list. We can provide it. We can provide it by email, and I'll follow up on that. Considering that it's also a growing list for all of that.
Le mot de la fin?
Le mot de la fin? Okay.
Yes.
So thank you, everyone, for attending this webinar. Thank you so much for the technical questions that were asked. They were very relevant, and it helps also tailor this technology. And we just thank you for your time. And if you want to reach out for the P-phaser, Hamza or I, and we're going to reach out also individually, but our emails on the website of WSP, or you can find us on LinkedIn also. And for Julia, the FREDsense, again, they have a website, so you can reach out to them as well if you need an immediate answer to any of your questions. Thank you again. Thank you for the presenters. Thank you for your time. And we're looking to hear more about anybody that wants to use the FREDsense or the P-phaser. Thank you, everyone.
Thank you.
Have a nice day.
Thank you so much.
Bye-bye.
Bye.